Okay, my turn.
Lee.
Right. So omeprazole, biovillager, and PCR. So kind of returning to omeprazole, which I talked about a few presentations ago. So just to start, so omeprazole is a protein pump inhibitor. Again, it just decreases stomach acid. So like acid reflux, GERD, which is gastroesophageal reflux disease. It's just like a chronic form of acid reflux. And any other conditions involving stomach acid, along with erosive esophagitis, which includes stomach acid in your esophagus. So in this, we have biovillager oxygenases, I can say that. So BVMOs, so they are just kind of a class of enzymes used. So they have oxygen, just molecular oxygen as acceptors, their cofactors FAD, and their electron donor is NADPH. And then cyclohexanone monooxygenases are a type of biovillager monooxygenase. So they're, oh, wow, I didn't even finish that. Okay, kind of messed up. But so their natural substrate is cyclohexanone, and they have a wide range of enzymatic abilities. So they're able to oxidize more than 100 non-natural substrates, and they have great regio, chemo, and stereo selectivity. So they're really desirable for processes. So they're able to do sulfoxynation and oxidation of nitrogen, boron, and selenium compounds, along with epoxidation of alkenes. So basically, they're just really versatile enzymes. But in this study, in this paper, they didn't do what they desired yet. So now we ask, like, what about omeprazole? So in this paper, this is the important part of omeprazole, the sulfoxide bond right here. So they started with omeprazole that didn't have this double bond to oxygen right here. It just was just a sulfur right here. So they called it omeprazole sulfide, and they were able to use an enzyme that made it into S-omeprazole, which is with this sulfoxide right here. So it was like just a really kind of revolutionary synthesis and implication of enzymes. So this is kind of the process they went about using. Again, so they started with this omeprazole sulfide, used the NADPH, the proton donor, or the electron donor, and used the O2. And they were able to make this S-omeprazole. So they used enzymatic asymmetric sulfoxidation using the molecular oxygen, which is the oxidant. And it was a promising green chemistry approach to the chiral sulfoxide production, just like it's seen here. So basically what this research did was it took an enzyme that did a bunch of different reactions in nature, and they trained it to do something unnatural, and they were really successful at it. So it's kind of different from before, where we had an enzyme that we knew what it performed and we wanted it to perform a specific already set in stone reaction to make a certain drug, Januvia. Here we don't even have that reaction. They're training this enzyme to do a specific reaction to make S-omeprazole.
Can I say something real quick, Emily, about
Yeah,
that
please.
figure? That so I just want everybody to notice that you may not have seen chiral centers at sulfur before like this. You know what I mean? Where the lone pair on sulfur is basically holds its configuration, so you can have a sulfoxide that's chiral, and in this case S. But that is sort of a funny thing about sulfoxides, is that they have four different groups if you include the lone pair, and they're configurationally stable. So if you make the S form, it like is the S form, and it sort of doesn't racemize into the mixture, if you follow me. So I don't know, people, I'm sure you probably haven't seen that before, but it is true that that structure is chiral, and it is also stable and doesn't just interconvert to the other isomer.
Okay, thank you. So this is just kind of a area diagram that highlights the mutations that we made. I don't really want to spend time on this, but yeah, it's kind of similar to the figure used in the last presentation in a way. It's just organized a little differently. So I kind of want to talk about how the mutations were made. So PCR, polymerase chain reactions, are a way to introduce foreign genetic material into bacteria that can eventually convert it to DNA, which will eventually convert it into protein that'll be isolated. So the simplified steps are to make the plasmid, introduce bacteria, and replicate as they find ideal mutations. So there's kind of a specific approach that they use for this. So this is a simplified, just visual of PCR. So it's a really exponential reaction. You're making copies, inserting primers, which are just synthetic molecules in high concentrations that help like mutate at a specific spot. So you're replicating each side of the chain. You're adding you're adding like a corresponding opposite side of that chain, and you're just copying each side every time. So it's really exponential. You're going to make a lot of these copies. So quick change is another process they use. I'm pretty sure these are like instructions, so I like this part.
Yeah.
So basically what they're doing is they're putting like, basically they know where, if they knew where to make the mutation, this is the process they would use. This is kind of introducing like where the primers are put, I believe. And they're copying.
Those little X's are the site of the mutation. The primers, if you go up a little bit, the primers are the little things with the arrows.
Okay.
Yeah, right there. Yeah. So so here it's a little weird, different because they have two primers in there, which actually normally people would just do one at a time. But yeah, that's
okay. Good. So this circle just kind of shows the plasmid in a way, and each spot in between these mutations is kind of the gene that you're mutating, is how I've understood it. So what they used in this paper was error prone.
Can I say one more thing, Emily,
no,
that
please.
I know you didn't know, but it's kind of like a neat little thing about how this quick change process works, is that when you do this, you're basically making some version of the plasmid that has the gene for your protein that has a mutation in it. But then there's a little bit of like leftover so-called wild type or unmutated plasmid. So it's like a mixture. And there's a really easy, tricky way to get rid of the plasmids that don't have the mutation, because the plasmids that you start with, you harvest those from bacteria. So you can use bacteria to not only make proteins, but to make DNA for you. So you harvest the plasmids from living bacteria, and then that's the circle at the beginning with the two different colored circles over there. Yeah. So that comes from a living thing. The mutant plasmid is purely synthetic. That's made by an enzyme in a test tube. So it's funny that bacteria that you get from DNA that you get from bacteria have a bunch of enzymes that add methyl groups on the DNA, and the synthetic DNA does not have those methyl groups because it was not born inside of a bacteria. And there's an enzyme that cuts DNA, but only if that DNA is methylated. So there's an enzyme that's the DPN1 enzyme on step 2 that cuts any DNA that came from an organism, but it won't cut DNA that's synthetic. So you can degrade the unmutated version of the plasmid and keep only the mutant version of the plasmid. Do you see what I mean? Yeah, I know all of you. A lot of these techniques are probably very foreign, so I would just want to applaud Emily for the like heavy lifting that she's doing right now to like talk about some of these practical matters.
Well, thank you. I'm trying. I don't know if I'm coming across well, but these are just like kind of short rundowns of these.
Yeah, yeah, this is very nice.
So what they used in this paper was error-prone method. So with error-prone, they're randomly inserting mutations through a specified gene where like the random part indicates random places being made. So here you can see that they're using manganese instead of magnesium. So magnesium is the ideal metal to use in this case, and manganese is not. So that's kind of a weird implication. And they basically like make mistakes by choosing the wrong NTP, which is nucleotide triphosphate, so GTP, ATP, NETP. And the mistakes are called mistakes because they don't mimic the parent chain. So I'm pretty sure these little like orange dots here are implicating like mistakes or mutations in the gene. So basically PCR is able to copy and amplify certain fragments, and we call these fragments genes, between two of the established primers. So this is another exponential process, and Scott even said it's used for criminal identification investigation. I thought that was
Yeah, that's true. Like, so when people get like biological material from like a crime scene, they use PCR to like amplify um, DNA from that material, and then they can compare that sequence to like either a database or like I guess a database or yeah.
right. So this was another part of the process that I found through. I don't even think this relates to this paper now that I'm thinking of it, Scott, but I just saw this in a different presentation. But what I was able to derive from this was that the plate assays were able to significantly improve screening of the mutant libraries.
Yeah,
But I don't think that does this come from this paper, Scott?
it totally does,
Okay.
and I'm glad you're showing this. So, do you know what you're looking at?
You know what? I tried very hard to understand this, and so these were
Yeah.
certain colonies, and these were other certain colonies. So I'm guessing this was colonies of the BVMOs with the thioester sulfoxidation. So with it having the double bonded oxygen, and without is how I understood it. They make a
Let's
lot of colonies
see.
with it.
Let me read it real quick. A and B, preliminary test A and B of a real mutant library screening plate. Preliminary test A and B and a real, okay. Plus and minus, rose and colonies a bit, yep. Oh, I see. Okay, okay. I think plate, okay. Well, the first thing to know, which is kind of interesting, is that so first of all, these are like agar plates, and on plate A and B, there are 6 bacterial colonies, which you can see as the little white spot. And each of those colonies has a different version of a potential enzyme that can do that sulfur oxidation, right? So this is how they're looking for, they'll make a whole bunch of mutants, and then they'll be like, okay, we're gonna take those mutant DNAs and put them in bacteria, and then we're gonna array all those bacteria that have a different mutant form, and we're gonna array them on this plate, which is sort of like in C. So C is actually the interesting one because there's tons of them, right? And each bacteria, it has a unique version of the gene that they're hoping does the reaction. Now, the readout is whether or not there's like a clear area around the colony or not, right? So how they did this is they put that. Can you just scroll up to the reaction again, real quick?
Mhm.
Okay, that's perfect. So the omeprazole sulfide, that molecule there, that molecule, I think you can appreciate, is a lot less water soluble than the sulfoxide product, the so the omeprazole on the right, because it's just more nonpolar. So what they did was they put the omeprazole sulfide, they just put it in the mixture that they poured the plate from. And because it's insoluble in water, it made the plates basically hazy. You know what I mean? Like if you add something that's insoluble to like an aqueous thing and then it solidifies, it'll be like opaque.
Mhm.
But if the bacteria can convert it to the sulfoxide, it clarifies because it becomes more soluble.
Okay.
Do you understand what I'm saying?
Yeah. So, like, if you see the dots on of the colonies, that's like it's not really dissolved.
Yeah. So go back now to the plates. They're in, so see how like some of those dots have like a clear halo around them?
Like this.
Yeah. No, like that one doesn't have a clear halo.
Oh, this
That
one
one
does.
does have a clear halo around the colony. That means that that bacterial colony has a version of the enzyme that can solubilize the omeprazole sulfide, presumably by converting it to the sulfoxide.
Okay.
So that colony is of interest. And so they would then pick that bacterial colony
Hmm.
and say, you have a mutant copy that I'm interested in.
Okay.
Yeah.
Very cool.
Yeah. It's super fast, right? Because there's no limit on how big they could make these plates. They could probably screen thousands and thousands this way. Yeah.
Okay. Very cool. So,
Sorry, I'm like shouting. I just got like a little excited.
no, I
I'll ask a
appreciate it.
question.
What?
Can I ask a question?
Yeah.
How do they get like what is the technique of plating like this? Like how do they make like the perfect dots? Like how does that happen? Because I feel like whenever I've plated something, it's been like a swipe and it's like all messy and like
Yeah.
do you know how that
Robot.
happens? Oh,
Yeah, this is definitely a robot.
interesting.
Yeah. Yeah. Because I don't know if you remember, Emily, I don't, I certainly don't, about how many colonies they sifted through, but they probably did thousands and thousands and thousands, and it was just a robot that has all these like little pins, and it would just pick them up and put them on a plate and
Okay.
then grow the plate. Because see how they're so perfectly spaced and stuff?
Yeah. So
Well,
too
they're
perfect.
fairly perfectly placed. Yeah. That's not from a person. That's definitely from a robot. Yeah.
Okay.
Yeah.
All right. So in the end, so this just shows, like it says here, the locations of the activity related to the amino acids um, found using like the error-prone method. So in the end, the final successful enzyme process used oxygen and glucose, and those were used as sustainable co-substrates, which like had water as a byproduct, making this a really cool like green reaction, and it was very promising approach to the S-omeprazole production. And that's kind of the end of what I had to say.
Can you go to the data slide real quick? The table of the data.
Sure.
And can you just tell people about the properties of the good, the best one?
And try-
Particularly, I think the EE would be of interest.
So anything, I'm guessing any of these with the 99% EE is the best because it like basically 100% shows the S configuration.
Yeah.
I mean, obviously, like these are pretty great too. This is horrible. This is the absolute worst. But like
Yeah.
these are fantastic. So I guess any of these.
And then the other thing that they're measuring is specific activity. Do you know what that is? That's kind of a weird enzymology term.
I feel like it would have to do with if it was able to, if the enzyme was able to work on the desired substrate only, is how I'm interpreting that.
That's exactly right. Its specific activity is sort of like the amount of catalytic activity per gram of protein. So see the units up there at the top?
Mhm.
It's U per gram. So U just usually means a standard amount of product formation per time. So how much omeprazole sulfoxide do you get like per minute per gram of protein? So the bigger that number is, that means that the enzyme is just is a better catalyst for that particular reaction. So
Clearly this last one.
yeah, the last one is definitely the winner because it has a specific activity of 30 units per gram, which is the highest one, and it maintains that high EE of the product. Yeah.
And these are all of the mutations that happen to make this ideal enzyme.
Yeah, it's quite a lot, right? And of course, like there's tons of mutations that they made that were either bad or didn't do anything that they just ignored because they had that nice way to see which ones were working by generating that like little clear halo around the colony.
So Scott, like, I do have a question on this. So like these are identical, these changes here, I guess, except for right here, these two lines.
Yes.
So what
Yeah.
happened? Why are these the same? Like, isn't once this is changed to lysine, like hasn't that already been changed to lysine? Or?
Yeah. It looks like the change for every row versus the parent, which would be the row above it. The change is bolded, and anything that hasn't changed is not bolded.
I see. Okay.
So yeah, so if you just pick one, if you look at like 5, right? So 5 has 5 mutations. It's got and then when they changed when they changed 5 to 6, they just changed what the mutation was at position 433, but everything else is not changed.
Okay.
Yeah. Yeah.
All right. So is there like any more questions on any of this? This is kind of a lot and kind of a tag team presentation, but
Yeah. So I'm sorry I interrupted you so much, but
no, I appreciate it.
I think it was my idea to make you talk about that, and then I was like, oh, it's like so new, and it's like actually like a lot of material. So
No, I appreciate it. Thank you. Yeah.
any questions for Emily? Can I talk a little bit about the coronavirus? Yeah, so you might know about the remdesivir, which is the nucleoside analog that is like it shows a little bit of activity against the people that have bad cases of the virus. There's a clinical trial right now, which is a little bit funny. It's omeprazole, as you know, is one of these acid reflux disease molecules that people take for that. And how it works is you probably know about histamine, right? Because you probably associate histamine with like getting hives. If you would have like an allergic reaction or like hay fever, you would take like an antihistamine like Benadryl. So histamine is a molecule that your body makes, and then when you take an antihistamine, you're trying to block the effects of your body's histamine, which is released from these like granules from like mast cells or whatever. So Benadryl being an antihistamine, what it does is it binds to the histamine receptor, blocking normal histamine from binding, and it keeps that receptor off. So in Beth's sort of signaling sense, it's a receptor binder that keeps it in the off state. So that's just called an antagonist. But there are two different kinds of histamine receptors. There's the H1 receptor